Welcome to the Deep Dive, the show that extracts the most important nuggets of knowledge from a mountain of sources, giving you those surprising aha moments that truly make you well informed. Today, we're diving into one of biology's really big questions, one that connects abstract ideas to, well, actual physical stuff. Where exactly are Mendel's abstract hereditary factors located in our cells?
That's a great place to start, because back in 1860, Mendel's factors, they were just this revolutionary concept, right? Something he figured out from his pea plants. He couldn't, actually point to them under a microscope. The idea was solid, but the physical reality, still a complete mystery.
Exactly. So our mission today is to pin down that physical basis of inheritance. We're moving from those theoretical factors to the real structures we now call chromosomes. We'll explore how genes sit on them, how their movement drives inheritance patterns, and maybe even look at some surprising ways they add it. Well, they don't always follow Mendel's classic rules.
That's right. We're going on a journey through some key discoveries, understanding some pretty unique inheritance patterns, like sex linkage, delving into how genetic information gets shuffled around, and then, really importantly, looking at what happens when chromosomes don't behave quite as expected. By the end, you should have a much clearer picture of what's happening at the chromosomal level.
Okay, so Mendel gave us the what, these hereditary factors, but the where was missing. And finding that where started not just with thinking, but with looking, right? Through microscopes in the late 1800s, what were they seeing?
Yeah, this is where cytology, the study of cells, really took off, mainly thanks to better microscopes. So between the 1870s and 1890s, scientists could actually watch the intricate dance of mitosis, how cells make identical copies, and meiosis, that specialized division for making sperm and egg cells. And they started noticing something really striking. There were these amazing parallels between how Mendel's factors seemed to behave and how chromosomes behave during reproduction.
Ah, okay. And this is where the pieces click together. Around 1902, Sutton and Boveri, they saw the connection.
Independently, yeah. They realized that chromosomes, just like Mendel's factors, come in pairs in our regular body cells. And during meiosis, when you make gametes, these pairs, the homologous chromosomes, they separate, just like Mendel said alleles segregate. Then fertilization brings pairs back together. That connection, that parallel behavior, led straight to the chromosome theory of inheritance. It was a huge leap.
Right. So the core idea is that genes aren't just floating ideas.
Exactly. Mendelian genes have specific fixed physical spots, we call them loci, along the length of chromosomes. Think of it like, for ages we talked about heredity like a recipe, but we didn't know where the recipe book was, or that it was organized into chapters at specific addresses inside our cells. That's the insight it's physically mapped out.
And the mechanism. It makes so much sense when you visualize it. It's the chromosomes themselves doing the segregating and assorting during meiosis. So, like, when homologous chromosomes pull apart in anaphase of I, that stage where they separate, that perfectly explains how the two alleles for a gene end up in different gametes, right?
Precisely. One allele physically moves one way, the other allele moves the other way, because they're on chromosomes that are splitting apart.
And the independent assortment.
That comes from how those chromosome pairs line up randomly at the cell's middle metaphase before they separate. That random alignment explains independent assortment for genes on different chromosome pairs. It's the physical reason behind that classic 9.3.3.1 ratio Mendel found in dihybrid crosses. You can picture those pairs shuffling randomly. It leads directly to all the genetic variety.
It's really elegant how the cell biology explains the genetics. But okay, that works for genes on different chromosomes. What about genes on the same chromosome? Or on the special sex chromosomes? Things get trickier there, I bet.
They do. And this leads us straight to Thomas Hunt Morgan and his fantastic choice of experimental organism, the fruit fly, Drosophila melanogaster.
Ah, the fruit fly. Why them?
Well, they breed like crazy, hundreds of offspring fast. A new generation pops up every 2 weeks or so. Plus, they only have 4 pairs of chromosomes, and they're easy to tell apart under the microscope. Just perfect for genetics.
So Morgan's got his flies, and then he finds something unusual.
He does. He finds this one single male fly with white eyes that was totally different from the normal or wild type red eyes. This was a mutant phenotype. And this one fly was key because how its white eyes were inherited was weird. It didn't quite fit the standard pattern.
How so? What did he do?
He crossed a normal red-eyed female with this unique white-eyed male. The first generation, the F1, they all had red eyes because a red is dominant, standard enough. But then he bred those F1 flies together to get the F2 generation. He saw the expected 3-to-1 ratio of red eyes to white eyes, but, and this is the kicker, all the white eyed flies were male. Every single one.
Whoa. No white-eyed females at all. That's Definitely not simple Mendelian inheritance. That screams sex is involved somehow.
Exactly. Morgan figured it out. The eye color gene had to be connected to sex.
Hmm.
Specifically, he deduced it was located on the X chromosome, and there was no corresponding eye color gene on the Y chromosome. So think about it. A male only has one X. If he gets that one X carrying the recessive white eye allele, boom, he's got white eyes. There's no second allele on the Y to mask it. We say he's homozygous for that gene.
Hemizygous, meaning just one copy, essentially.
Right. But a female, she has two X chromosomes. She needs to inherit the recessive allele on both of her X's to actually have white eyes. One dominant red allele on one X is enough to give her red eyes.
That fits perfectly. And it ties right into how sex is determined chromosomally, at least in mammals like us, the XY system. Females XX, males XY.
Yep. And the Y chromosome is tiny compared to the X, carries way fewer genes.
So conception is basically a 50-50 shot from the sperm.
Pretty much. All eggs have an X. Half the sperm carry an X, half carry a Y. If an X sperm wins, you get XX, a female. If a Y sperm wins, you get XY, a male. Simple as that.
And isn't there one specific gene on the Y that's super important? The SRY gene.
Ah, yes. The sex-determining region of Y. Crucial. Its presence triggers the development of testes in an embryo. Without a functional SRY gene, even if the chromosomes are XY, ovaries develop instead.
Okay, so genes on either sex or chromosome are called sex-linked, but the X has way more, and they're not all about sex determination, right? Like the fly eye color.
Exactly. The X chromosome is loaded with genes for all sorts of traits, many totally unrelated to sex determination, like color vision in humans. The Y, on the other hand, has very few genes, mostly for male characteristics, and they pass directly from father to son only.
Which leads us to things like X-linked recessive disorders, because males are hemizygous.
They're much more likely to express the trait. They only need one copy of the recessive allele inherited from their mother, because there's no second allele on the Y. Females usually need two copies, one from each parent, to show the trait. So we see these conditions way more often in males.
Like red-green color blindness?
Classic example.
Yeah.
A father with it passes the allele to all his daughters, making them carriers if they get a normal allele from their mom, but never to his sons. A mother can pass it to either sons or daughters.
And there are more serious ones too, like Duchenne muscular dystrophy.
Sadly, yes. Affects about 1 in 3,500 males. Causes progressive muscle weakening and hemophilia.
The blood clotting disorder. Famous in European royalty, wasn't it?
Yeah, Queen Victoria was a carrier, and it spread through royal families because of, well, royal intermarriage. It highlights how these single gene changes on the X can have huge historical impacts. Thankfully, today we have treatments like protein injections.
It's fascinating. But there's another wrinkle with X chromosomes in females, isn't there? They have two X's, males have one. How does the cell balance that out? It seems like females would get a double dose of X gene products.
Ah, nature's clever solution. X inactivation. In female mammals, very early in development, one of the two X chromosomes in almost every single cell gets randomly shut down.
Shut down? How?
It condenses into this tight little structure called a bar body. You can actually see it under a microscope. Most of the genes on that inactive X just aren't expressed, they're silenced.
And it's random which X gets inactivated, the one from mom or the one from dad.
Completely random in each cell lineage early on, which means female mammals are actually mosaics. They're made up of patches of cells where the maternal X is active and patches where the paternal X is active.
Wow, a mosaic. So literally a patchwork quilt at the cellular level.
That's a great way to put it. You are literally a blend of both your parents expressed differently across your body's cells.
That explains tortoiseshell cats. The fur color gene is on the X, right? So you get patches of orange fur where one X is active and black patches where the other is active.
Exactly. That's the visible manifestation. And in humans, say a woman carries a recessive X-linked gene that prevents sweat glands from forming. She won't lack sweat glands entirely. She'll have patches of skin with normal sweat glands and patches without them. A living mosaic.
That's truly mind-bending. Okay, so we've covered genes on different chromosomes, genes on sex chromosomes, but what about different genes that happen to be on the same non-sex chromosome?
Right. This brings us to linked genes. The thing is, you have way, way more genes than you have chromosomes. Each chromosome is like a long string carrying hundreds, even thousands of genes. And genes that are physically located close to each other on the same chromosome tend to get inherited together. They don't assort independently like Mendel saw with his P traits because they're physically stuck on the same piece of DNA.
Okay, let's clarify that. Linked genes are on the same chromosome and tend to stick together. Sex-linked genes are just genes on a sex chromosome, X or Y. They can be linked to other genes on that same sex chromosome, but the terms mean different things.
Perfect distinction. And Morgan demonstrated this too with another classic fruit fly experiment. This time he looked at body color gray versus black and wing size normal versus tiny vestigial wings. These genes were not sex-linked.
So what did he do this time? A test cross again?
Exactly. He took females that were heterozygous for both traits. They had one chromosome with gray body normal wings and the other with black body vestigial wings, and crossed them with males that were homozygous recessive for both black body and vestigial wings.
And based on linkage, you'd expect mostly offspring looking like the original parental combinations, gray normal and black vestigial.
That's mostly what he saw, yes, which confirmed the genes were linked. But, and here's the next puzzle, he also got a smaller but significant number, about 17% of offspring with new combinations. Gray body vestigial wings and black body normal wings. These are called non-parental phenotypes, or recombinants.
So the linkage wasn't absolute. How could you get new combinations if the genes are stuck on the same chromosome?
This is where genetic recombination comes in, specifically through a process called crossing over.
Ah, crossing over, the great reshuffler. How does that work again?
Okay, so during prophase I of meiosis, those paired up homologous chromosomes get really cozy. Specialized proteins actually make cuts in the DNA of chromatids from different parents, non-sister chromatids, and then swap corresponding segments before rejoining the breaks. It's like they literally trade pieces of their ends. So even though the genes for body color and wing size started on the same chromosome, a crossover event between them can create a new chromosome with a mix of alleles, say, gray body and vestigial wings.
But crossing over breaks the linkage sometimes. And the further apart two genes are.
The more likely a crossover event is to happen somewhere in the space between them, leading to recombination. Genes very close together, they almost always stick together. Genes far apart on the same chromosome, they might recombine so often they look unlinked.
And this frequency of recombination, that led to gene mapping, didn't it? Morgan's student, Sturtevant.
Alfred Sturtevant, yes. Brilliant insight. He reasoned that the percentage of recombinant offspring you see is a measure of the distance between the genes on the chromosome. It's like if 2 points are far apart on a string, there are more places you could cut between them. He proposed that 1% recombination frequency equals 1 map unit of distance.
So you could build a map. Like if gene A and B recombine 10% of the time, and B and C recombine 5% of the time, and A and C recombine 15%.
Then you know the order must be A, B, C, with B closer to C than to A. You can build a linkage map, an ordered list of genes along the chromosome based purely on recombination frequencies from breeding experiments.
That's incredibly clever. Are those maps accurate?
They give the right order, which is amazing. The distances are approximate because crossing over isn't perfectly random everywhere on the chromosome. And really distant genes on the same chromosome can recombine 50% of the time, just like unlinked genes. Today, we can sequence the entire genome and get the physical map of the exact number of DNA-based pairs between genes. But linkage maps were the first step, and conceptually spot on.
And thinking bigger picture, all this shuffling from independent assortment and crossing over, why is it so important?
It's the engine of genetic variation. It creates new combinations of alleles in every generation. This variation is the raw material that natural selection acts upon. Without this constant reshuffling, populations couldn't adapt nearly as effectively to changing environments. It's fundamental to evolution and the diversity of life.
Okay, so we've seen the elegance, the mechanisms,
Yeah.
but things can go wrong, right?
That's right.
Big changes to chromosomes.
Yes, unfortunately. Large-scale alterations can happen, maybe due to errors in meiosis or damage from radiation or chemicals. In humans, these are often serious, frequently leading to miscarriage. Plants, interestingly, tend to tolerate these kinds of changes much better.
What's the main way chromosome number goes wrong?
It's usually due to something called non-disjunction. This is basically when chromosomes fail to separate properly during meiosis. Either the homologous pairs don't separate in meiosis the first, or the sister chromatids don't separate in meiosis the second.
And the result is sperm or eggs with the wrong number of chromosomes.
Exactly. You get gametes with one extra chromosome, N+1, or one missing, N1.
So if one of those fuses with a normal gamete,
The resulting zygote has an abnormal chromosome number that's called aneuploidy.
aneuploidy.
Yeah.
And there are names for specific types.
Yep. If you're missing one chromosome, it's monosomy, so you have 2N1 total. If you have an extra one, it's trisomy, 2N+1 total. Most aneuploidies in humans involving the larger chromosomes are lethal early in development.
But one major exception is Down syndrome, trisomy 21.
Correct. That's the most common human aneuploidy that allows survival. It's caused by having 3 copies of chromosome 21 instead of 2, so 47 chromosomes total. Individuals typically have certain facial features, shorter stature, often heart defects that can be corrected, and developmental delays. But it's a spectrum, and many people with Down syndrome lead fulfilling lives, hold jobs, and contribute greatly.
Is there a link to eternal age?
There is a significant correlation. The risk increases as the mother gets older. For instance, under 30, it's maybe 1 in 2,500 births, but by age 40, it's closer to 1 in 100. The reasons are complex, likely related to the aging process in eggs, which are held in meiosis site for decades.
Okay. What about having entire extra sets of chromosomes, not just one extra?
That's called polyploidy, so having 3 sets, triploidy 3N, or 4 sets, tetraploidy 4N. This is actually quite rare and usually lethal in humans and most animals, but it's super common in plants.
Really? Like our food crops?
Oh yeah. Bananas are often triploid, wheat is hexaploid, 6N. Strawberries can be octoploid, 10. Polyploidy has been a major driver of plant evolution and agriculture.
Fascinating. What about aneuploidy of the sex chromosomes? Is that as severe as having an extra chromosome 21?
Generally, no, it's less severe. There are a couple of reasons. One, the Y chromosome has very few genes. Two, any extra X chromosomes beyond the first one are simply inactivated and become bar bodies. The cell compensates.
So what conditions result?
Well, you can have XXY, which causes Klinefelter syndrome in males. They're usually sterile, might have some female-like body characteristics. Intelligence is often normal, but sometimes slightly subnormal. XYY males are typically taller than average, but otherwise fairly normal, no well-defined syndrome. XX females, trisomy X, are usually healthy, maybe a bit taller, fertile, though sometimes with learning disabilities.
And the only viable monosomy in humans?
That's Turner syndrome, where individuals have just one X chromosome, X0.
Right.
They are phenotypically female but sterile because their ovaries don't mature properly. Intelligence is usually normal, and estrogen therapy can help with development.
Okay, so that covers a number of problems. What about changes to the structure of a chromosome, like breakages?
Right. Chromosomes can break, and the fragments can be lost or reattach incorrectly. You can have deletion where a piece is just lost entirely.
Like Cri-du-Chat syndrome.
Exactly.
Yeah.
That's caused by a specific deletion on chromosome 5. Leads to severe intellectual disability, a small head, and that distinctive cat-like cry in infants. Very serious.
What else can happen after a break?
You can have a duplication, where a fragment gets attached as an extra segment to its sister chromatid or homologous chromosome, or an inversion, where a fragment breaks off, flips around, and reattaches backward, or a translocation, where a fragment joins a completely different non-homologous chromosome.
Translocation, like pieces swapping between different chromosome types?
Yes, often it's a reciprocal translocation, where 2 different chromosomes exchange fragments. A really significant example is in certain cancers, like chronic myelogenous leukemia or CML.
How does that work?
In CML, there's typically a reciprocal translocation between chromosome 9 and chromosome 22. Part of 22 breaks off and sticks to 9, and part of 9 sticks to 22. This creates an abnormally short chromosome 22 called the Philadelphia chromosome. Crucially, this translocation creates a new fused gene on the Philadelphia chromosome that codes for a protein that triggers uncontrolled white blood cell division leading to leukemia.
Wow. So even if no genes are lost, just moving them around can cause cancer.
Absolutely. The location matters. Moving a gene can put it under the control of different regulatory elements or fuse it with another gene, altering its function dramatically. While inversions and balanced translocations might not lose genetic material, they can still have devastating effects depending on where the breaks occur and what genes are affected. Deletions and large duplications are often very harmful or lethal.
What an incredible journey. From Mendel's abstract factors to the physical reality of genes on chromosomes, their dances in meiosis, the exceptions like sex linkage and linkage itself, and then the consequences when that structure or number goes awry.
It really is. We've seen how those abstract factors found their physical home on chromosomes, how sex chromosomes create unique patterns like X-linked traits and that amazing mosaicism in females, how linked genes aren't completely stuck together thanks to crossing over, and the really profound impacts of those large-scale errors in chromosome number or structure.
So this deep dive really gives you that solid foundation, right? Understanding the chromosomal basis of inheritance is just so fundamental for biology, medicine, evolution.
Absolutely. It connects so many dots. It helps explain everything from why certain diseases run in families to how new species might arise in plants. It's core knowledge.
So as a final thought for you listening, just consider the incredible complexity, the sheer precision needed for our genetic blueprint to be copied and passed down correctly generation after generation. What does this intricate dance of chromosomes, with all its built-in mechanisms for variation but also its potential for error, really tell us about how adaptable and maybe how fragile life on Earth truly is? Thanks for joining us for this deep dive.